All right, good afternoon, everyone. Welcome to this week's Iowa Learning Farms Conservation webinar. I hope you're somewhere safe and warm while the blizzard here in central Iowa rages on. Can hear the wind outside my window here. It's a great day to be on a webinar here somewhere warm, and we are really fortunate to have with us Dr. Adina Howe. Dr. Howe is an associate professor in the Department of Agricultural and Biosystems Engineering, a colleague, and I hope I can say friend as well in the department. And so we're really excited to hear her talk today on our agricultural microbial neighbors. So with that, I'll turn it over to Dr. Howe. If you have questions, please type those in the chat box as always, and get those to Adina at the end of the talk. With that, Adina, the virtual floor is all yours.
All right, thanks, friend. Definitely friend here. I'm going to try to put my face on the slides because I think it's kind of neat to see a more interactive seminar. And it's a pleasure to be here and to talk to you. And I'm looking forward to your questions and interacting. I'm trying to give a couple of brief stories about some of our research that our team does here at Iowa State, looking at microbiomes associated with agriculture. But first, let me talk a little bit about microbiomes. So a microbiome is a community of tiny living organisms. They could be bacteria, viruses like COVID, fungi, or other microbes that live in a specific environment. And these environments can be anywhere. They could be in the soil, they could be in water, they could be inside our bodies and our guts. And we, the field of microbiology, is increasingly aware of the role of these microbes in our lives. For example, for us to be healthy, we rely exclusively on bacteria in our guts to produce vitamins, such as B12. We wouldn't get B12 except for the breakdown of those enzymes by bacteria in our guts. They also help break down complex carbs for our energy. And we're also increasingly aware that it's not a single or even a few bacteria, but a community of bacteria that are working as a team to make these things happen for our health, and similar to our health, also healthy environments. So besides being in our bodies, microbiomes are also present in and on animals and growing plants. So these bacteria, fungi, or viruses are moving between these systems. They're an important facet connecting human health and agriculture as they're on these animals and plants. And in the time I have today, I'm going to tell you about two projects related to these microbiomes. And the first is in the area of animal production and manure management. And the other is how we're trying to rethink sustainable agriculture and the growth of bioenergy crops. I am not monitoring the chat till the end. Okay, I see that Dr. Helmer's put a comment in there, but I'll check out it at the end so that I don't get sidetracked. All right. I am proud to be an Iowan, and Iowan animal producers are also very proud to feed not only Iowa, but much of the world. And along with pig production, there is a lot of pig poop, and there is a lot of it. We're now producing many more animals and much denser populations. And so you have many more animals and many more pooping in one location. This poop, if you don't know, and many of you probably know a lot more about it than I do, gets stored basically in what I think of as a giant bucket underneath the barn. And when I joined Iowa State in 2015, these manure storage pits were having an issue. And what was happening in these manure pits is as the animals pooped through the flats in these barns, the poop in the bucket stored underneath would start to produce a foam that would come through the flats, making contact with the animals. And it was also releasing methane gas, which you may also know is highly flammable. And this could lead to damages and very high flammable fires, and of course, animal deaths and human deaths. And because this was happening, when I came on board, I was asked if I could help figure out what was going on from this foam. And the reason a microbiologist would be when asked about these foam is because this methane is produced by bacteria called methanogens. And so there was a direct link to the microbiomes. And so they knew that these pits, these foams, foaming pits were associated with methane. You can see that in that graph here, that the foaming pits had a lot more methane. We also knew that these pits could be characterized in different ways. You could have foam on the top, or they'd be crusty, or they wouldn't be foaming. And so what we did to try to figure out what was causing the foaming is we went out across the state of Iowa in a year, and we grabbed hundreds of samples of pits, and we classified them in 3 different states, like I said. And these states would be that they were either foaming, crusty, kind of like a biocrust, like kind of like looks like kind of dry dirt maybe, or no foaming, kind of what a pit should look like. So we classify them in these 3 categories, and under these 3 categories, we then measure manure characteristics, the manure chemistry, and also on my team, we would study the bacteria or the microbiome. Now, when we study the microbiome, and let me try to communicate this like in a very quick way, but it's pretty complex. But what we do is we basically fingerprint who is there. We're doing kind of a survey of the different bacteria that there are there and how many there are. And this is not too dissimilar than what you might have seen in crime scene investigation shows, where they're doing forensic investigations and they're trying to find a fingerprint of somebody who has left DNA that's similar to what's left at a crime scene. And they're looking at patterns and they're trying to map who's the most similar to those patterns. And that's what we're trying to do here too when we do these studies. We're trying to look at fingerprints of the different bacteria that are there, the abundance that are there, and we're trying to figure out what's similar in foaming conditions versus non-foaming conditions. And we could also do this for not just bacteria, but just general characteristics of those samples. Which samples are grouped together? And if there are patterns, we can start thinking, well, if we can understand those patterns, then maybe we can understand what's causing foaming. And so each of these dots on this figure here, which is a chemical fingerprint of the manure characteristics of those hundreds of samples we took all over Iowa, what we're looking at is which of those samples are close together. So each dot is a sample. The closer together they are, the more similar they are. And when we color the dots, you can see that the foaming samples have a unique fingerprint that looks like a group right here of orange that are put together. And the non-foaming also looks separate. So from that, we can say that foaming and non-foaming manures had unique chemical signatures. And we could even figure out what chemical signatures those are. And we could talk more about that if you want to. We can do similar things with the type of bacteria that we see in these manure pits. And so again, now instead of foaming and non-foaming, I have foaming here, bacteria kind of clustering together, then the crust samples, and then the non-foaming samples. And these are the bacteria. And we can also use the same fingerprinting technique for the methane producing bacteria very specifically. And you can see that these really separate into unique signatures. So based on these analyses that we do as microbiologists, we can say that the Iowa foaming manures and the non-foaming manures had unique microbial fingerprints. Now, we can take a look at these data, combine the chemical and the microbial data, and we can look for links of when one increases maybe chemically, another bacteria increases chemically, and we can start to connect some dots together. And real briefly, I can tell you that when we could find those links, and in foaming manures, we found that the microbial community was able to really efficiently do fatty acid conversion. And in non-foaming manures, they weren't so good at it. I want to take a minute here and step back from fatty acid chains to something that's maybe more relatable and talk about, when we talk about fatty acid chains, what we're really talking about is digestibility of carbon or fiber. So let me just step back and say what we think is happening in these foaming manures. At the time, so this is 2015 when I came on board here, something that was big in Iowa was ethanol production. And a byproduct of ethanol production is a waste product, maybe a waste product in this particular context, is distillers' dried grain. And this grain is made up of long chain fatty acids, highly fibrous. And because it was a waste product, it was being added to feed because it was a nice use of this product in an effective economic way. This increase of this grain in the feed caused the gut microbes in these pigs to change, and as they pooped, changed the content of the pits. And so what we think has happened is during the ethanol demand of the 2015 era, we increased these digestible grains. This changed the guts of the animals. It changed the content of the long chain fatty acids. This selected for different bacteria that caused methanogens to grow, and this caused the foaming. And so this is an example where we could really make some links on the microbiome perspective of adding to like understanding how to manage manure and give some direction for feed in the future. So this was my first story. And my second story, we're going to switch gears to a totally different area, and that's growing crops. So this is a soil underneath bioenergy plant production. And our team is really fortunate to be part of a larger center called the Center for Advanced Bioenergy and Bioproducts Innovation, CABI for short. And this is funded by the Department of Energy. And the goal of CABI is to develop economically viable and ecologically sustainable bioenergy crops. So growing bioenergy crops, unlike food, we don't want to take up the rich soils. We want to be growing this in marginal lands where we're not already growing crops that can feed people, but we are also trying to produce sources for biofuels and bioproducts. And so it's a bit of rethinking how we do agriculture because we have to grow them in totally different landscapes. The reasons you want to grow these bioenergy crops is we're trying to capture these petroleum-based industries and replace them with these bioproducts, mainly things like sustainable aviation fuel or high-value pharmaceuticals. And really, we're targeting hard to electrify sectors. When we're trying to produce these bioproducts, one of the biggest targets for these things like sustainable aviation fuels is lipids. And so you can get lipids from different things. You could take waste greases, you could take them from, you can convert them from wastewater. One of the other things you can get lipids from is plants. And so CABI, part of the center is to figure out how to grow plants in a way to provide these lipids that we aren't growing plants for now. So growing these feedstocks and developing these feedstocks is a large directive of our center. One of the crops that we've been developing in partnership with the University of Florida is sugarcane. So these plant breeders have been brilliant at engineering sugarcane, which usually produces sugar for food. Instead of producing sugar, they're telling the plant genes to then produce oil. So they're engineering these plants to produce oil. And they've been really successful at it. They can produce oil canes now that produce 70 times more than a sugarcane plant will by just changing the genes in the plant. The problem is when they plant these oil canes in the field, you can see that here. We have our wild type, which means the original sugarcane, non-engineered. And then we have two different variants of the bred oil canes with different genes tweaked on them. And you see they're wimpy in the field. So there's this yield drag from this engineering. And so this team came to me and said, hey, Adina, do you think that the microbes underneath are different between these plants? And do you think these microbes could help maybe this yield drag, or maybe it could also help these plants grow in different conditions under drought or with water limitations? And I said, I really don't know. And so what we did is we did an exploration project to figure it out. So I'm going to skip this slide, but if you're interested, I left it here so that you can take a look at it if you watch recording, and you can pause. What we're looking here is the amount of lipids. This tag is a acronym for the lipid that we're trying to target. How much more you get in these engineered than the wild type. So 0.06 is the wild type, up to 4 times here, or 4% dry weight here. And the different genes kind of gives you an insight into how they do this on the plant side. So what we did is we took these different plants and we grew them up on the same soil, greenhouse soils, and we took samples of the microbes attached to their roots. And we wanted to know if those microbes were different in those plants as they grew in the greenhouse for 3 months. And then we did the same thing, took these plants out and put them in the field and took the same root samples and characterized those microbiomes. And so what I'm going to show you next is the actual fingerprint of those microbes in these plants. So on this, one of the things of I think these webinars is learning how to read scientific figures. So I really tried to put something that was something we actually published here. On this fingerprinting map, what you see on the columns here are the 3 replicates. These are the sugarcane, the wild type, and then each one of these unique acronyms is a bred engineered oil cane. So there's the 17T, the 65, 66, 69, and 80 oil canes. Those are each plant's a sample from the root of each plant, a replicate. Each row here is a bacterial taxa or species. So this is a bacteria 1, 2, and there's hundreds of rows. And then I color them in by the observation of the numbers of those bacteria. Let me shut my face off here so you can see that a little better. So what you can see here is, maybe I can argue with you that there's unique fingerprints. This 1566, this 1566 column here, these 3 columns, they look different, right? They're green over here and red over here than the other variants. And that indicates that they're a different microbiome underneath. And this 1566 is the highest lipid producing plant that they've engineered. So something funny is going on. What's really interesting, this is the greenhouse samples, is when we go out to the field, you see that the 1566 looks largely the same. We have these green patterns and these red patterns, but everybody else, all the other oil canes and the wild type even, start looking more like that underneath. What that means, what the functions these are providing, that's where we are in the project right now. Our next step is actually to understand what these bacteria do. We can look at the genes that are in their DNA and figure out what they're doing. I can tell you early results suggest that these are secondary metabolites or stress responses of the bacteria, probably because the plants themselves are under stress. And then trying to connect that then to how we can use this information to help the plants grow or maybe be managed in a better, sustainable way in different stressful conditions. All right, I was asked to keep this short so you'd have time to ask questions. So in conclusion, agricultural microbiomes, I hope I've given you some insight on what the role that they can play in sustainable farming and ecosystem health. They are involved in nutrient cycling, plant health, and soil structure. They can help manage resilience and disease, not just in the agriculture, but in our bodies. We here at Iowa State and in my team, we think of them as a resource. They can be studied, understood, predicted, and potentially, if we do it right, managed. So those are some of the projects we're working on. I should also acknowledge the team. The job here isn't the research itself, but training the next set of scientists that's going to go out and really make the big differences. And I'm lucky to be a part of this team that's so wonderful, and also acknowledge the funding sources that have enabled this science. That's all the slides I have. I know there are more slides, but maybe it's question. I think the next step is the question time.
Let's go through those slides, and then
okay.
You can take a breath, and people can start typing in more of their questions. But we have our normal end of webinar slide. So if you watch this webinar live and are wanting to earn a CCA CEU, please email Elena Whitaker, ALENAW, at iastate.edu by 5 o'clock today. Include your name, the name you entered to watch the webinar, and your CCA number. And again, that's ALENAW at iastate.edu. Next slide.
I got you.
Okay, there we are. If you watch this webinar live and are willing to fill out a voluntary demographics survey, we would greatly appreciate that. Only one survey response is needed for all 2025 webinars. You can go to the site there, iowalearningfarms.org/survey, or the QR code that's on there. So again, if you've done it in 2025, thank you. If you're willing to, we'd greatly appreciate it. In the next slide, we'll highlight the webinar coming next week, which is Dr. Mike Castellano. It looks like if you can forward that slide. Talking about the Iowa Nitrogen Initiative and the new NFAC tool, Decision Support System for N Fertilizer Management from the Iowa Nitrogen Initiative. And then I think one more slide should be today's webinar and all previous webinars in the ILF webinar archive are online. You can go to the iowalearningfarms.org/webinars or to the Iowa Learning Farms webinars YouTube page. So we got some great questions here. So first, Adina, can you talk about how you get the microbes from the plant root? How do you separate root from the soil, and what distance from the root is impacted?
Right. Okay, that's a terrific question. okay, how we did it. Let's first talk about how we actually did it. So what we do is so in order to get the root microbiomes, we actually have to dig up the plant. So it's a sacrificial sample here. We can't keep checking in on the plant after because we have to dig it up. Actually, soil studies are generally difficult this way. Every time you take a sample, you have to poke a hole for lots of different reasons, not just from the microbial standpoint. We take the root up and then we shake off basically as much soils as we can. Then we go in and we take tweezers on the roots and we try to separate out just the roots. And it's a really laborious process of trying to find soil attached to the physical roots. And that's what we're calling the rhizosphere. This debate of what is the rhizosphere is discussed quite a bit in our field. And I think there's a consensus that there can be some flexibility in how you define it. We find the largest signal of who's interacting with the plants are going to be on who's actually on those roots. So the closer we can get at that scale, I think the better our results often are. Where does the rhizosphere then end, right? All these plants grow, and especially perennials, those roots have been touching that soil for a legacy of time. That's a great debate. I don't know what my recommendation is, except that we tend to just try to get against the plant as much as possible to try to hedge our bets. But you have to take into fact, if you're one of more samples, you might not have the time or the energy to do that tedious tweezering for all the plants. So, but in this case, we did.
Okay.
Because it was important.
Next one, was manure or commercial fertilizer used in any of the sugarcane work?
Okay, no, not in that work. Though I think as a part of thinking about sustainable practices at large for bioenergy crops, as a part of the bigger question, things like cover crops and different nitrogen sources are a part of that question, but not a part of this particular work.
Any information about that maybe highlights the differences in the biology between, dry cattle manure versus commercial biology products relative to corn production?
Okay, I'm trying to figure out what commercial biology products might be.
And it might be because there's another question here about kind of asking about if you're familiar with work done with rhizobiome and analyzing root rhizospheres and making predictive models to evaluate the effectiveness of biological products in soils.
Okay. I am broadly familiar with this work. There are a lot of companies, industry trying to figure out kind of the personalized medicine, personalized agricultural bit of microbiomes and their ability to help specific products grow. And historically, we know that soybeans and nitrogen fixers are our thing in microbes. So we have this idea that, yes, you can put beneficial. I am familiar with it. I can say from my expertise and my experience, it is really hard to make things stick. There are a lot of bacteria in the soils. They're very diverse. You take one oil cane field and another oil cane field, their bacterial can be they might only share 1% of the same bacteria, but they're all doing the same thing. And so the idea of putting something on and having it stick is still an area that we don't quite understand. That delivery is a ripe area for research and something we're interested in. So maybe that helps. So I guess I don't know much. I'm enthusiastic about it. I haven't heard a lot of momentum successfully in that application.
Okay. A second. How does the microbial community in the rhizosphere vary throughout the year? And in what extent fluctuation, or do you see fluctuations with precipitation, temperature, field management, so forth?
Yeah, that's a terrific question. It varies across seasons because the biology is going to be influenced by temperature, water, and the pH conditions of the soil. And so they're responding to the plant. So as the plant varies, you're going to see the rhizosphere vary as well. The biggest question is, I think we know that it changes. I think the biggest question is what can we do to make it change in the directions we want it to change? And that's where really the open questions are.
Okay. Got one on the foaming. And they were interested in how different feed profiles might affect foaming, and could different feed profiles produce more
Yeah.
methane and less hydrogen sulfide?
Okay. I'm not sure about hydrogen sulfide. So I bet Dan Anderson would know a little bit more about that. But yes, definitely different feed profiles will produce different. So there's the manure component of like when you mix it all together, and then there's actual the individual animal component. And I think in both cases, you would expect that just like what we eat causes us to fart differently, this would also affect the animals and the manure content. So
I guess the kind of in a follow-up with this, seemed to indicate that there was more foaming with the use of dried distiller's grain. And
yes.
has this influenced changes in feed rations at all that you know of?
Yes, I don't know if our study influenced it, but definitely the price of corn and ethanol demands influenced it, and that has changed the composition of what's going in the feed.
Uh, Somebody said they'd like to hear some more things about the new things your team has developed to allow us to study microbes and microbiomes, excuse me.
Yeah.
Maybe comment on how they might be used to learn some new things in the years to come. So where do you see the future of this work, maybe?
Yeah, that's a pretty thick question. And a great question. Thank you, Dr. Van Lykke. Um, I think the future of the work is figuring out how we can see patterns in an individual system and apply them broadly. Like, so how do we take the stuff we're seeing in an individual field, but then apply it to all maybe perennial grass growers? Because these incremental science things can help a niche, but we're really also seeking to understand like the theory, the fundamentals of how they work together, how they persist, and that'll really increase what we can do. Technologically, the DNA sequencing has changed everything about how we get our getting treated as well. That'll be very exciting to use these as we learn how to use the newer methods of dealing with large data sets, being able to look at our ability to survey in larger ways and be able to sample with partnerships with many of you here, many different sites to understand patterns. That'll be also really exciting.
We talked a little bit about, some of the fluctuations, but there's a question here about from what you've done for microbiome, could you sort of rank the factors that maybe influence the microbiome in the soil? Texture, weather, plants being grown, tillage, fertilizer use? Is there a general
Oh, geez.
consensus?
Yeah, that's a good question. It varies quite a bit. The plants are definitely a big part. For example, whether you're along a root or not in a bulk soil, that's an easy place to see differences. But we actually see a lot of spatial variation, and that's going to be deal with the kind of the package of soil characteristics, the water, temperature, and those are bigger. Those are bigger than the rhizosphere just as place, or else we'd be in, maybe I would argue would be in really have trouble doing it. That's a terrific question. I am hesitant to rank it because I think I might get it wrong because it varies by the question we've studied, but definitely sites, climate, sites, soil location, and then plant scale. Maybe
Okay.
I'll start there.
Your friend and colleague, Dr. Anderson, asked that question, so he's going to hold you to it. No,
Yeah, he will. I know he
yeah.
will. Good.
Okay. Is it possible that whatever you might learn can be used in our efforts to improve the microbial health of our soils for normal crop productivity?
I think absolutely. I would say yes. If I couldn't, I wouldn't be doing it. I think it might not be like we put it, maybe a lofty goal is being able to deliver a microbe that could just fix things. Probably not going to happen. But things like maybe we should rotate this crop out because it selects for a specific group that then can't be, once it's selected for, can't be brought back to its diverse natural conditions, or in a state of drought or extreme weather, can't deal with that stress as effectively as a more diverse community. I think those pieces can help us think about how to manage the crops in the system that we have them in. I think there is also hope in delivery of microbes. Let me not be too cynical on that. I just think the industry has really honed in on an easy solution there. But just like a magic pill is not going to fix everybody, I think the same thing applies. And soil is even more complex than that. But yeah.
I think, that's interesting. It makes me think of, that how can we learn from that to make a more resilient system so that it can respond to those drought and drought or wet conditions. Another one, was there any correlation found between soil type and microbiome found? I think you kind of answered that, but
yeah, not in this particular study, but in many studies there are.
Okay.
Mm-hmm.
Okay. I think that pretty much gets most of the questions there, Adina.
Terrific.
Again, if folks are wanting the CCA CEU, please email Elena Whitaker, ALENAW @iastate.edu by 5 o'clock today with your name, the name you entered to watch the webinar, and your CCA number. And then also, please tune in next week to Dr. Mike Castellano talking about the NFAC tool. And thank you, Adina, for taking the time.
Yep, thank you for your attention.
Blizzard. Yeah, we had somebody's passion and brilliance. Many thanks.
Oh my gosh, let me just end the day now.
Better than that.
Let's go home. So, yeah, let's go.
All right. Thank you so much for participating, and thank you everybody for attending. Take care.